Hello and welcome to today's video where we are going to understand a very important topic of biology, especially pertaining to molecular genetics and molecular biology, that is linkage and recombination. So, first we have to understand the key phrases and terminologies pertaining to this topic. First is linkage, second is recombination, third is chromosome, fourth is recombination factor, fifth is crossing over, sixth genes, and seventh alleles. We'll understand these terms as and when needed while studying this particular topic. So first, it's prudent to give you a basic idea of chromosomes and how the genes are located on the chromosomes. These are the chromosomes. Let's say inside humans, we have 23 pairs of chromosomes. Why don't we say 46 chromosomes? We say 23 chromosomes. It would have some specific reason. Each of the chromosome in our human body has a set or a pair. That means, let's say this is chromosome number 2. So this chromosome number 2 has one chromosome from the maternal side and one chromosome from the paternal side. So this is the basic introduction to chromosome. Now, in each chromosome, there are certain regions where the genes are there. Let's say the gene for our height. Let's say the gene for our height is located in chromosome number 2 and it is in this position. That means that at this particular position, in each of these two chromosomes, we will have the gene for height. So here we have a gene for height, and here also we have a gene for height. So we have understood chromosome, we have understood genes. Now let us understand alleles. Now height, according to Mendel, can be of two types. Number one is tall, and number two is dwarf. So let's say here we can have a gene for tall, and here we can have a gene for dwarf, or we can have the vice versa, or in both the cases we can have genes for tall or gene for dwarf. Okay, so what does crossing over mean? If you have studied the cell cycle and cell division, especially the topic of meiosis, you will understand that in process 1 of meiosis, what happens is that these chromosomes, a part of these chromosomes get exchanged. Let us say that these particular parts are exchanged in process 1 of meiosis 1. So that means that this particular part will now be attached to this chromosome and the brown part will be attached to this chromosome. So this is what crossing over generally means when we talk from the point of view of meiosis. Now let us understand the meaning of linkage and recombination. Before understanding the meaning of linkage and recombination, it is important why these terminologies came into molecular biology in the first place. See, Mendel gave three laws of genetics. Law of inheritance, that they are actually known as law of inheritance, where the third law was law of independent assortment. What it meant was that the expression of one gene or one allele rather was independent of the other gene or allele. Let's say we have a dihybrid cross, tall plant, dwarf plant, yellow seed color, and green seed color. That means tall can merge with green and dwarf can merge with yellow, thereby forming a new character or a new progeny. Okay. So why are we calling it recombination? Because in none of the progenies rather, we are getting the exact same characteristics as are present in any one of the parents. So that is why we say it is a particular case of recombination. That is, the characteristics are recombined. Now, what Mendel said is that there's a 50% chance of this particular characteristic, and there's a 50% chance. That means that if we have TT cross TT, T can merge with this T or can merge with this T. So the first capital T can merge with the first small t or can merge with the second small t. So there's a 50-50 chance that it will merge with the first small t or with the second small t, according to the Mendel's law of independent assortment. Okay. So after that, we will be understanding linkage and recombination. Let's say we have a chromosome here, and in this chromosome, let's say we have chromosome number 5. In this chromosome number 5, at this particular position, let's say position 7, we have that gene for height. So in both these chromosomes, we will have gene for height. And let's say in the position 9, we have genes for hair color. So in both these chromosomes at position 9, we will have the gene for hair color. Now, linkage means that height and hair color, the genes for these two characteristics are present in the same chromosome. Linkage means the two genes are present in the same chromosome, and if they are present in the same chromosome, there is a greater possibility that they will be present in the progeny at the same time. Let's say we have the height of a tall height here and hair color black. So if they are present in the same chromosome, then there is a very little chance that they will get mixed up. That is, in the progeny also we will get tall statured, black colored persons. But that will also depend on number of factors. Most important is the distance between these two genes. So you have got a basic idea of linkage. Now let us again understand what recombination means. Already recombination, I think you have got a glimpse of this particular term when we were discussing law of independent assortment. Recombination actually means different phenotype, different phenotypes in the progeny as is present in the parent. So let's say we have two different chromosomes here. This is chromosome number 5 and this is chromosome number 11. So actually in this situation, in chromosome number 5 we already have height and hair color, and in chromosome number 11 let's say we have a gene um, for memory. It's an imaginary gene for memory, and we can have different phenotype, two different phenotypes, sharp memory and dull memory. So if we have the parents tall, statured, and black, and we have the parents as sharp and dull, there is a high chance that the progeny will have black color, tall, statured, but there is no guarantee that the uh, progeny will also have a sharp memory or the progeny will have a dull memory. So we cannot ascertain clearly whether the progeny will have a sharp memory or the progeny will have a dark memory, dull memory, because they are present on different chromosomes. Let's say we have a parent who is tall statured, has black hair and sharp memory, but the progeny can be black colored, tall statured, dark memory because the gene encoding memory is present on a different chromosome than the gene encoding height and hair color. So this is the main idea about linkage and recombination. Now we'll understand what is non-parental recombination, what is parental recombination, and one of the most important questions of this topic, that does being on the same chromosome guarantee 100% linkage or not? Let's say we have a chromosome here, and in this chromosome here we have a height and here we have a hair color, and let us say this is chromosome number 5 again. So what actually happens is that during meiosis there is a situation of crossing over where the end parts of these chromosome the end parts of these chromosome are actually transferred or mixed up. So the end parts of these chromosomes are mixed up. That means if the end parts are mixed up and on the end part only we have the hair color gene, so there's a high possibility that there can be recombination despite being present on the same chromosome. Because let's say we have here brown and here we have black, but during meiosis this orange will be shifted here and this red will be shifted here. So obviously we can have a tall statured brown hair colored or progeny and a short statured black hair color progeny, although none of the parents show such characteristics. So despite being present on the same chromosome, we can have recombination. That means we can have different characteristic sets than the one present in the parent. Fine. So we have understood the topic of linkage and recombination. So what determines the difference between linkage and recombination, the possibility of recombination, although being present on the same chromosome, it is the distance between the genes. Distance between the two genes that are present on the same chromosome. Okay. So this distance is actually measured in a unit called Morgan, named after the founder of this concept, Thomas Hunt Morgan. Actually, this scientist, Thomas Hunt Morgan, worked with Drosophila melanogaster, a very important organism as far as molecular biology is concerned. Just like your E. coli is a very important organism, so Drosophila melanogaster is also a very important organism. It is commonly named as, let me just write the name for you, Drosophila melanogaster. I'm also not very accurate with the spelling. Melanogaster. So this is the one with which Th Morgan actually worked, and they are commonly referred to as the fruit flies. There are various reasons for Th Morgan to work with this particular organism. The most important one is that the breeding time is very low, and that this Drosophila melanogaster produces a large number of offspring in a very small amount of time, and the variations are easily seen in them. So actually, he worked with white-eyed, yellow-bodied females with brown eyes, red-bodied males, and gave the concepts of linkage and recombination, and it led to the eventual concept of gene mapping. And gene mapping was developed by Th. Morgan, later modified and enriched by his student. But what his student did was that student introduced the concept of Morgan as the distance between the two genes. But we don't generally use the unit Morgan. We use a smaller unit called centimorgan. 1 centimorgan is equal to 10 to the power minus 3 Morgan. And for information, 1 centimorgan is equal to 1% recombination. So this is what is called a recombination frequency. Recombination frequency means how many offsprings are there possessing recombination. So from this recombination frequency, we can get an idea of where the genes are actually located on a chromosome or on different chromosomes. Let me just give you a basic image of Drosophila melanogaster gene linkage map. This is a historical figure as it was the very first gene mapping that was done in molecular biology. So here this entire diagram was done by Thomas and Morgan and Stuttgart. So here you can see the wild type and mutant type. The wild type is denoted as capital A, the mutant type by small a. This is a miniature diagram of the chromosome, and inside the chromosome you can see the DNA coiled up. Now in we have a map like this where we basically locate various genes, like see we have 0.0, 13.0, and more is the distance between two genes, more is the chance of recombination, and lesser is the distance between two genes, lesser is the chance of recombination, or higher is the chance of linkage. So gene mapping is actually used in medical science for the for the diagnosis, and also to some extent manage the condition of cystic fibrosis. So, cystic fibrosis is a genetic disorder in which there is a problem with the CFTR protein. The CFTR is actually a protein that helps in salt and water regulation in our body. So, actually, due to a defect in this CFTR gene, rather not a protein, but in this CFTR gene, what happens is that the person loses too much water through their sweat, and as a result, the mucus and other substances of the uh, gallbladder and pancreas becomes too much thickened, and ultimately what happens is that the patient suffers from unintentional weight loss and eventual respiratory failure due to problem in the bronchi and the bronchioles. So the patient develops infection and chronic respiratory failure due to a condition called bronchiectasis, which is associated with cystic fibrosis, and eventually dies due to infection of the lungs. So for the diagnosis and treatment of cystic fibrosis, this gene mapping is frequently used, and cystic fibrosis is generally diagnosed by the saliva or the sweat test. So I hope this video is clear to you and you have got a little bit of information about linkage and recombination. Thank you.